Hello, everyone. It's very nice to welcome you to the Biochemical Society and Portland Plus webinar. And this is, as you might be aware, a part of the Biochemistry Focus webinar series. Topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support your career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. And you can go to the website for more details. I'm Urszula McClurg, and I'm very pleased to welcome you to today's topic, which is investigating the microbiome. And we have two exceptional new PIs talking to you about this. And this is a part of a dedicated early career research program of webinars where you can see scientists who are the future of their fields. And we are delighted to provide this opportunity today to Drs. Cartmell and Patrick Moynihan. And they can share their work with the molecular biosciences community. So before I hand over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar. So not at the end of each session, but please do send your questions in as the talks happen and as you think of them. If you have a question, please type it in the question box as shown in the image on the screen, starting with who your question is for. And we will try to answer as many as possible at the end after Patrick's talk. So our first invited speaker today is Dr. Alan Cartmell from the University of Liverpool. Alan did his bachelor's in biochemistry at Newcastle University and then completed both an MRes and a PhD under the supervision of Professor Hallie Gilbert. And then he undertook a 2-year postdoctoral fellowship at the La Jolla Institute for Allergy and Immunology in the laboratory of Professor Derek Zions at the US. Afterwards, he returned to the UK to Professor Gilbert's laboratory, where he worked on elucidating how members of the human gut microbiota catabolize plant glycans from the human diet. He's currently a lecturer at the University of Liverpool, and he has just secured a welcome CDA with which he's moving to a senior lectureship at York University. And his group has a focus on exploring how sulfated glycans, such as glycosaminoglycans and sulfamucin, are metabolized by both the HGM and the host itself. So, Alan, we are very much looking forward to your talk.
Thanks for the introduction. So I'll get on with it. So my current focus of research is on carbohydrate sulfatases and their critical roles in the metabolism of sulfated carbohydrates. In particular, I look at the roles of these enzymes from bacteria that are present in the human digestive tract. The human digestive tract is essentially an outside external surface of the body. Humans can be thought of as hollow tubes. On the outside, you've got an impenetrable barrier, the skin, but on the inside, this has to interact with the environment and allow exchange of materials such as food. And it also interacts with bacteria from the environment, and these bacteria colonize the gut. And in response to this, the epithelial layers of the digestive tract have to protect themselves to be able to do these roles adequately. And the way they do this is by forming a gel-like polysaccharide called mucin that lines all the epithelial surfaces of the digestive tract. And this mucin changes in its type and composition depending on the part of the digestive tract we're looking at. And of particular interest to me is colonic mucin, as this becomes heavily sulfated. So mucin is around 80% carbohydrate by mass. By the time you get to the colonic mucin, about 10% of this is sulfated, or 10% is sulfate by mass. And so this sulfate is critical for the functional roles of this mucin in the colon. And you actually don't see sulfated mucin elsewhere, or not to a significantly high level. And the reason for this is we have bacteria colonizing this mucin layer, and this bacterial relationship is actually symbiotic in most cases. These bacteria provide around 10% of our daily calories, train and regulate our immune system, and produce molecules that affect our cognitive function. And they use the mucin layer as a colonization factor and nutrient source, but it's set up so that degradation of the mucin layer by the bacteria is slower than the production, and you actually have this sterile mucin layer that acts as a barrier. And the reason why this layer in the colon is sulfated is because the bacterial load is at its highest. Once you get to the distal colon, there are more bacteria there than all the other microbiomes in the body, and there are the same number of bacterial cells as there are somatic cells in the human body. And so this extra layer of sulfation, this extra sulfation adds a layer of complexity that slows down the metabolism of this mucin and maintains a symbiotic relationship. So host production of mucin matches bacterial degradation. However, in disease states, you see dysbiosis and degradation of the mucin layer by the symbiotic bacteria exceeds production, and you get greater penetration of the bacteria, which leads to inflammatory bowel diseases. Carbohydrate sulfatases are a class of enzymes that are essential for the bacteria of the human gut microbiota to access sulfated mucin. So in a normal system, these carbohydrate sulfatases are needed just for the bacteria to colonize and perform their normal symbiotic roles. However, in disease states, we see that these are overactive. Carbohydrate sulfatases are currently classed into 4 families, of which the S1 family comprises 90% of all sequences and is the only one that's been implicated with carbohydrate metabolism. So S1 family, only kind of carbohydrate sulfatases that have been found, and these are heavily present in bacteria of the human gut microbiota. These S1 sulfatases all have a conserved fold. It's an alpha beta alkaline phosphatase-like fold, and they all use a specially encoded nucleophile. It's non-genome encoded to form our glycine. So during the translation of sulfatases, a cysteine or serine in this consensus sequence is converted to a catalytically competent formyl glycine. And so we've actually known for a long time that elevated carbohydrate sulfatase levels of the gut bacteria are implicated in ulcerative colitis. And we've known this since around 1995. But then more recently, a mouse model came along that allowed us to show this in more detail. And what it showed is in this mouse model that is susceptible to developing severe colitis, when it was gavaged with wild-type bacteria that had active carbohydrate sulfatases, it developed severe colitis. However, when it was gavaged with a sulfatase deficient strain, it did not develop colitis. And you can make this sulfatase deficient strain by knocking out the enzyme that forms that critical formyl glycine nucleophile. And this is just the mouse model in a bit more detail. You have in the top panel, the genetically acceptable mouse model, and you gavage it with PBS and you don't see any inflammation. You take a wild-type mouse with wild-type bacteria, you see no inflammation. If you take the genetically susceptible mouse with wild-type bacteria, you see severe inflammation. If you take the genetically susceptible knockout mouse with a sulfatase null version of the bacteria, you don't get inflammation. And if you complement the maturation enzyme that activates the sulfatases back in, you then get severe colitis. And so we've got quite a good in vivo model for looking at carbohydrate sulfatase dependent colitis. And so with all this context in place, we looked at bacteria from the human gut microbiota called Bacteroides. And these are significant carbohydrate degraders of both host glycans and dietary glycans in the human gut microbiota. And we picked members that had a significant number of carbohydrate sulfatase genes, and we looked for their growth on colonic mucinoligosaccharides purified from pigs. So these colonic mucinoligosaccharides are heavily sulfated, and these gastric mucinoligosaccharides are not sulfated. And what we were essentially looking for was robust growth on these sulfated carbohydrate oligosaccharides. And fortunately for us, the model organism, which is called Bacteroides theta iota omicron, so B theta for short, grew quite well. and that had 28 S1 carbohydrate S1 sulfatases. We didn't know they were carbohydrate sulfatases because the S1 family can do a range of activities other than just desulfate carbohydrates. And so the beauty of this organism is that it's genetically tractable. And so it was genetically tractable, and the highest number of S1 sulfatases in the human gut microbiota, and had been shown in the mouse model to drive colitis. And so the next thing we did was we cloned every sulfatase from Bacteroides, theta, iota, or micron, and we screened them against a panel of commercially available oligosaccharides to start assigning activities to these enzymes. And we looked, there's 82 subfamilies, and so we looked at what activities we could see by subfamily, and you can see it starts to break down into specific subfamilies targeting specific sulfated oligosaccharides. We also took a mass spec approach. This is where we used our clonic mucin oligosaccharides in very small amounts and treated them with different enzymes. So here's 6 enzymes. Here what we're looking for is the absence of a sulfated structure. Its absence in the mass spec chromatogram tells you that the enzyme has worked on it and it's no longer there. And so this combination of using commercially available sulfated oligosaccharides, mostly mono and disaccharides, and purified clonic mucin oligosaccharides allowed us to ascertain the activity of around 16 enzymes out of 28 that B theta has. Some of these were new activities within a subfamily, and some of them, such as the S120 and S116 and S146, were the first activities ever described for these subfamilies. With the biochemical information in hand, we then set about making genetic mutants, and then using these genetic mutants to explore how the loss of a particular sulfatase that we know the biochemical activity for affects growth. Our first mutant was the enzyme that activates sulfatases, the anaerobic sulfatase maturation enzymes. This is the one that introduces that special nucleophile, the non-genome encoded nucleophile. And what we can see is on gastric mucin oligos that are not heavily sulfated, barely sulfated at all, we don't see a growth defect. But on clonic mucin oligosaccharides, we see a significant growth defect. But what was particularly interesting to us is that out of all the genetic knockouts that we made, some of them were compound deletions. So you can see here we made 8 times sulfatase knockouts and 10 times sulfatase knockouts. But the only mutant that had a significant effect was the loss of this 1636 gene, which desulfates galactose and mucin. Knocking out this single sulfatase had an effect similar to knocking out every single sulfatase through the loss of the maturation enzyme. So we can see in this 8 compound deletion, there is no significant effect on growth. And in this 10 times knockout, we did see an effect on growth, but we could mitigate this by complementing back in this 1636. And you can see this 1636 has no effect on gastric mucin oligosaccharide growth. So it is a sulfated, it is a specific defect associated with metabolizing sulfated glycans of mucin. So this was very interesting, as we found a single sulfatase that appears to be a keystone step, but it was quite puzzling that none of those sulfatases seem to have a significant defect in vitro. And so we've got this model where we know that in disease states, carbohydrate sulfatase activity is elevated. And now we have this model for Bacteroides that are drivers of this disease and the main metabolizers of carbohydrates in the colonic environment. We can see there's a single sulfatase that appears to control the metabolism by this organism. So essentially, we've got a keystone step. And could we target this keystone effect with inhibitors to maybe treat ulcerative colitis? So we've got quite a nice direction forward where we're going with the project. But something was a bit puzzling is, although 1636 was the only enzyme that had any significant defect genetically in growth, but well, when we knocked it out in growth, there were two other enzymes that could do the same job. There was another enzyme called 1622 that could also work at desulfating the same linkage as 1636, and 4683 could also desulfate the same linkage as 1636. And that linkage was O3 sulfate on galactose. So we had three enzymes that could all desulfate galactose at the O3 position, but only one of them had a significant effect on growth. And so we were lucky enough to be able to solve the structures of all three of those enzymes. Two of them were in the same subfamily, so 1636 and 1622 were in the same subfamily. 4683 was in a different subfamily. What we can see from the structural work was that 1636 had a really high affinity reaction with the hydroxyl O2 of the galactose, so this hydroxyl here. This was lost in 1622 in favour of accommodating a much bulkier N acetyl group that occupied that position. Everything else was the same, so it was just this discerning feature at the C2 carbon. 4683, on the other hand, had a much more open active site. And this enzyme actually turned out to be an endo-acting enzyme as opposed to these two exo-acting ones. And this data was borne out in binding assays. So this is differential scanning florimetry, where we use the product to look at our substrate to look to see if it stabilizes the enzyme, and that's a proxy for binding. And we can see when we use galactose, 1636 is stabilized, but the other two enzymes aren't. 1622 was stabilized by this N-acetylgalactosamine, which has this much bulkier group at C2 as opposed to just a hydroxyl group. And it was stabilized by this, but not by galactose. And then once you get to 4683, it's stabilized by a disaccharide, but not the monosaccharide, indicating that it needs to engage multiple subsites to bind. So you've got this enzyme that has a really high affinity for the O2 hydroxyl of galactose. This one seems to be tailored for a different substrate, 1622, although it can desulfate galactose at the O3 position. Its binding is really, really weak and is probably not physiologically relevant. And this one targets a very specific structure in an endo fashion. Another feature of 1636 that was unique is that it's actually on the bacterial cell surface. So we have this high affinity interaction for O3 sulfated galactose at the cell surface that appears to be critical. And looking at the S120 family, you can actually see there's, we did both sequence alignments and phylogenetic distributions, and we can see that the key activity clusters into a couple of groups. And you can also see this more unusual activity accommodating that bulkier sugar splinters off over here. And so we can start to classify other enzymes in the family and what they do. And we can see there's a strong preference for 3S-gal and also a conserved 3S-GalNAc activity, which is that bulkier sugar. And so we've got this story that O3 desulfation of colonic mucin oligosaccharides is critical for Bacteroides of the human gut microbiota to metabolize mucin. And these O3 sulfated galactose residues tend to be terminal decorations. So this desulfation of galactose is essentially the first step that occurs in the desulfation of colonic mucins, and it's a critical step. We then moved into in vivo mouse competition experiments, and indeed, the 1636 mutant was unable to compete with wild type B theta, and you can see the relative abundances here. But what was also interesting was that we saw compound effects when we used deletion of the 3S-gal sulfatase and the 6S-glucanac, suggesting that although in vitro these sulfatases didn't have a significant defect, in vivo they are important, suggesting that there are actually maybe two steps that you can target to slow mucin degradation in these bacteria. And so we use an integrated strategy to try and elucidate what these enzymes do. We start off with the enzymology, moving into the structural biology, so we know what their substrate target is and how they bind it, and we then transfer that into genetic systems where we can look at how the loss of the organism affects growth. And we're currently trying to get this into mouse models. And we've been doing some small molecule screening. And this is some of the small molecule screening we've done. And this was against so this is the critical sulfatase that we identified in the Bacteroides species. This sulfatase was the one that had a compound effect in vivo, but had no effect in vitro. And these enzymes have exactly the same sulfate binding site, but they differ massively in their carbohydrate binding regions. And what I really want to show here is that this is the same fragment library against two enzymes. And you can see that the hits, so each one of these each black dot is a molecule that's been screened against the enzyme. And we can see that this line sets 50% inhibition, and we have 3 really good hits here, as well as several other 50%, which for a fragment library is quite good. But none of these hits transfer over into the S111 enzyme. And so we can see that not only do we have specificity for the carbohydrate between these enzymes, but we can see there's in the same fragment library, there's no overlap in the molecules that inhibit them, which is great for a drug screening project and strategy and developing lead compounds which can be modified, hopefully, into small molecule drugs. So the summary of this work is really carbohydrate sulfatase are incredibly specific enzymes. De-sulfation of mucin or glycans at the bacterial cell surface is critical for chronic mucin or glycan utilization by Bacteroides theta iota micron. And this is actually conserved in Bacteroides species of the human gut microbiota. So this is hopefully a general theme. And carbohydrate sulfatases are drivers of disease. And obviously, I just want to acknowledge everyone who's been critical to this work: Anna Sophia Luis, who's in Gothenburg, and Eric Martins in Michigan, as well as Miriam Chiswick and Tristan Barbaron at in Roscoff in France. And I just want to thank everyone for listening.
Thank you very much, Alan. This was fantastic. As a reminder, all the questions will be at the end, but please type them in the chat as they come to you. So with that, we will now move on to Patrick Moynihan from the University of Birmingham. Patrick completed his PhD in December 2013 and moved to the UK as a postdoctoral researcher at the University of Birmingham in the laboratory of Professor Besra. From there, he obtained a BBSRC Future Leaders Fellowship to study mycobacterial peptidoglycan recycling. Patrick subsequently received a BBSRC David Phillips Fellowship in mid-2019. It was, I think, one of the last rounds of these fellowships, in fact. And that is when his research group started to work on the proteins and enzymes that govern the microbacterial cell wall. Since then, Patrick has had a lot of success. He's now a lead on a big Wellcome Discovery Award, so we're privileged today to hear from 2 Wellcome-funded scientists. And with that, Patrick, over to you.
Thanks so much. A small point of clarification, actually Liz Lowe leads on the Wellcome grant.
Apologies.
It's all right. It's been a team effort, but she's definitely the lead. All right. Thanks for the kind introduction. Thanks for those of you who've come today. My name is Patrick, and as said by Urszula, I work at the University of Birmingham. And my group has a long-standing, medium-standing, I guess, interest in mycobacteria and their cell envelope specifically. So just by way of providing some context, I get my pointer working. There we go. We are interested in mycobacteria for a range of reasons, but chief amongst them is this graph here. So this is from the World Health Organization, and the arrow you see here is pointing at tuberculosis. It's the 13th leading cause of death for all mortality worldwide. It is a major global catastrophe. We see about 1.6 million deaths annually due to tuberculosis, about 10 million new infections every year. And we really, COVID made this much worse, and so progress has really been set back by many, many years as a consequence. So this is something that really we need a lot more work on. We need better vaccines, and we definitely need better medicines and diagnostics. So that's kind of the human side of why we're interested in mycobacteria. But biochemically, I became interested in this organism because of this structure here. And this is the cell envelope of mycobacteria. So for those of you who work in bacterial systems, you'll be familiar with some elements of this structure. Those of you who are not, I'll try and give you a whistle-stop tour of it. So like almost all bacteria, mycobacteria have a phospholipid membrane, which I've tried to artfully draw as a gray box here. But mycobacterial's phospholipid membrane is quite a bit different. It's predominantly comprised of an unusual lipid called phosphatidylinositol monosaccarides, or PIMS for short. And so these are basically just monosylated glycolipids that make up the majority of the envelope. Attached sometimes to the cytoplasmic membrane, you get this molecule called lipoarabinomannan, which is effectively a PIM on steroids. So they take the mannose backbone and hyperextend it. I've drawn a truncated variant here. And then that'll become lipomannan. And then occasionally that lipomannan will get appended by a large D-arabinan domain, and it becomes called the mature molecule called lipoarabinomannan. Now, importantly, the D-arabinan here is actually D-arabinofuranose units, and D-arabinofuranose is a pretty rare sugar in all of biology. You only find it in a few places, predominantly amongst mycobacteria, but it does pop up in some other species here and there. Now that's the kind of envelope or the membrane associated structures, but really the star of the show is the large covalent cell wall complex. And so like almost all bacteria, this starts with the peptidoglycan saccharase. Being mycobacteria, they have to do everything a little bit differently. So mycobacterial peptidoglycan is a heavily modified structure. So a lot of the things that you know about, say, E. coli or bacillus or staph don't hold true here. And then attached to the mycobacterial peptidoglycan is this large branched polysaccharide called arabinogalactan. It's so named for two domains, has a D-galactan backbone. Again, that's in the furanose configuration, so D-galactofuranose. And appended to that is a D-arabinan domain, similar to what you find in lamb, though as far as we're aware, what our data would show is that they're not by any means identical. So for those of you who work in plants, maybe you'll find a similarly named polymer in plants, but structurally they're completely different. So what you know about plant enzymes and whatnot won't hold true in this system. And then finally, esterified to the termini of arabinogalactan are these large lipids called mycolic acids. And these are really kind of a hallmark molecule in mycobacteria used in diagnostics. They pull them out of like mammoth skeletons and stuff. So Del Bezer here at Birmingham's done some work that way. They give mycobacteria their characteristic waxy outer coating. So if you grow Mycobacterium smegmatis on agar, for example, it looks like candle wax. If you poke it with a loop, it'll crack like candle wax. They're just incredibly hydrophobic. Intercalated into the covalent lipids will be a host of glycolipids. I've only drawn trehalose mycolates here, but there'll be a range of other lipid species found in that outer membrane. So mycobacteria aren't really gram-positive, they aren't really gram-negative, they're gram-indeterminate, but they do have this true outer membrane. Now, if you can imagine, bacteria are at their heart engineers, right? And there's a famous quote, and the person who said it's eluding me right now, but the dream of a bacterium is to become two. Right? And so they want to grow and divide. They need to grow and divide, and they need to do that while withstanding their own interior turgor pressure, which can be greater than that of a car tire. And if you've ever gotten a nail in your tire, you know that's bad news. And so these engineers have to be able to take their cell wall, cleave it without blowing up. They need to be able to do that to insert large, say, envelope spanning secretion systems. They have to do it just for simple growth. And all of that takes a host of enzymes that act on virtually every major covalent linkage in this structure, or at least presumed to act on that. Now, we know a lot about how peptidoglycan is degraded. And we actually know a fair bit about how mycolic acids are removed. We don't know anything really, or very little, about how arabinogalactan is turned over in mycobacteria, and almost nothing about how lipoarabinomannan is turned over in mycobacteria. And this is really important because, of course, the covalent structure of the cell envelope isn't the whole picture. Mycobacteria do produce a capsule. It's predominantly alpha glucan, but interspersed in that, we know are fragments from the cell wall. We see pieces of D-arabinan, pieces of lipoarabinomannan, and these are really immunogenic. They're important for how hosts sense the bacteria. And so this really got us to the question, which is, how do mycobacteria cleave their arabinogalactan? How do those fragments that end up in the cell envelope actually get there? Okay, So just to drill down on arabinogalactan specifically, just a little bit more to give you a sense of the complexity of the molecule we're dealing with here. So PG stands for peptidoglycan down here. So you have PG on one end, mycolic acids on the other end. And in fact, as far as we're aware prior to our work, there's only ever been a single enzyme that degrades any component of this structure identified. So every colored unit on here is a new linkage between two sugars. So the backbone of galactan, for example, is 1516 beta galactofuranose. And so Shen in 2020, they identified an endogalactanase. So this is an enzyme that cuts within this galactan chain. It's not a particularly active enzyme. It takes a long time to do that job, but it is fairly specific. Conversely, no enzyme has ever been described or named that works on this complex structure. However, as I mentioned, there's some evidence they exist. I've shown you we know there are fragments in the capsule. There's also data going back all the way to the 1970s and a little bit into the 1960s showing that these enzymes almost certainly exist in nature. So the bottom, these are two works from Catani in 71 and 72. The bottom one, I had to get someone in the British Library to go to the microfiche stacks and actually print out the paper for me and send me a scan of it. These papers, effectively what the groups did was to take arabinogalactan purified from bacteria and throw them at, from mycobacteria, pardon me, and throw them at some soil microbes and then do kind of old school chromatography fractionation of the soup of enzymes these bacteria produced. And they came to this one fraction that would reproducibly degrade the D-arabinan backbone of arabinogalactan. Now, unfortunately, in the 70s, they didn't really have the tools to identify that enzyme. And in fact, groups have been trying ever since then to figure out what these enzymes are because they are clearly performing what should be a very crucial role in the biology of the bacteria. And yet no one's been successful in doing that so far. So this really raises the questions, where are these enzymes, right? Should we be able to look at the genome and look at that? Well, about 40% of the mycobacterial genome has no annotated function. And for people who are attending a Biochemical Society webinar, I probably don't need to convince you that genomic annotation is completely reliant on prior biochemical assignment, right? We need biochemistry before genomics has any meaning. And so the question becomes, how do you find new activities if there are no characterized homologs? And this is where I teamed up with my friend Liz Lowe, who's a lecturer at Newcastle University. And Liz and I, we first met on Twitter, had some back and forth, and then we actually met at a Microbiology Society conference and got talking about this problem, about why we can't find enzymes that break down mycobacterial arabinogalactan. And Liz introduced me to the wonder of the microbiota. And Alan's done a great job of introducing this, so I'm going to try and be very specific about the points relevant to our work. And this slide's a little bit out of date, the paper's from 2013, but the message is absolutely still true. So on the x-axis, we have the number of different enzymes that degrade carbohydrates. On the y-axis, we have the total number of different families of those enzymes. So y-axis is diversity, x-axis is total number. And this was at the time all known bacterial genomes. You'll notice is the top right quadrant, so the bacteria with the most and the most diverse enzymes is almost entirely dominated by these Bacteroides, right? These bacteria that live in your microbiome. And so Liz introduced me to the concept that these could be potent screening tools. They can degrade just about anything. And the really beautiful thing about these bacteria is the way in which they encode the enzymes that break down carbohydrates. And I'll come on to that in a little bit, but they become a very great screening tool. So really, in terms of fitting into a microbiome webinar, I really want to pitch the idea that Liz convinced me of, that the microbiome is a really great screening tool. Okay, so what did we do? Well, we took 14 members of the human gut microbiome and we challenged them to grow on arabinogalactan as a sole carbon source. The idea being that if any of these bacteria could actually grow, they would upregulate the genes that they use to grow on that substrate. Then we could identify them using omics approaches, RNA-seq or proteomics, and we could use that information to go fishing for these enzymes in mycobacteria, assuming that nature hasn't messed about too much and that there will be homologs of these in the mycobacteria. So we did the experiment, the first step being growing these bacteria on arabinogalactan, and we were a bit surprised, but not surprised, I guess, that almost all of the bacteria did in fact increase in biomass when they were asked to grow an AG. So this tells us that in that polysaccharide, there are enzymes, or sorry, in those bacteria, there are enzymes that can break down that polysaccharide. but that doesn't really tell you what part, right? I mentioned that there are two components to arabinogalactan, arabinin and galactan, and because there'd already been some information about galactan, we really wanted to know about arabinin. So I learned that from Liz that Bacteroides are a bit like toddlers. They're really messy eaters. So if you want to know what they've been eating, you can kind of just look in the area around them, in this case, the culture supernatant. And if they're breaking down a polysaccharide, they're going to produce monosaccharide derivatives of that polysaccharide that will leach their way into the media. And this can tell you what they're eating. So for those of you who've never run any kind of HPLC experiment before, I'll quickly go over how this works, because there's a couple of these traces through the talk. Basically, it's a chromatogram, so we're separating out a complex mixture on a solid support. We separate the molecules based on their physicochemical properties, and we can compare that to standards, so in this case, arabinose and galactose. So that means anytime we see a peak above here, it's a galactose-containing peak. Anytime we see a peak above here, it's a arabinose-containing peak. And it allows us to quickly figure out what's going on in this complex mixture. And so what we can say with certainty is that almost all the bacteria were growing on the D-galactan. Now, in retrospect, this is not that surprising. D-galactofuranose is actually fairly widespread in biology. You find it in LPS, in capsular polysaccharides. But thankfully, only one species, or at least one species, I should say, was able to grow on the D-arabinan. And this was this bacterium called Disgonomonas gattii. So Liz and I got Matthias Trost involved, who's a proteomics expert at Newcastle, and collected the biomass, gave it to Matthias. And Matthias then analyzed the most highly upregulated proteins in Disgonomonas gattii. I'm skipping over a little portion of this story for the sake of time. There's a little bit about how we got to pure D-arabinan, but we'll leave that for you to read if you want to, or ask me later. And this is where the concept of a polysaccharide utilization locus comes in. So most Bacteroides organize their genome into these discrete loci that degrade a given polysaccharide. So for example, if you're eating bread, they'll turn on their mannan locus. If they're eating cellulose, they'll turn on the cellulose locus. Or if they're eating D-arabinan, they're going to turn on the D-arabinan locus. And what Liz and I got really excited about in these data is that this pull that was identified by proteomics, almost all of it are proteins with no known function. It's like catnip for biochemists, right? We've got all of these exciting new enzymes and we don't know what they do. So I'm going to take you just through two of the families we've identified so far. The first is the DUF2961s. At the same time we were doing this work, some lovely work came out of a group in Japan that found similar results. The papers in JBC, it's Kashima et al. I encourage you to go and have a look, but I'll go over some of our data on this project. So there's 3 DUF2961s in this pull, and we're initially a bit skeptical about them because based on sequence analysis, this was before AlphaFold when we started this, they don't look like GHs. In fact, there was already one structure from a structural genomics consortia in the PDB, and it didn't look like a GH. So we didn't really know what it would be. Nonetheless, between Liz and my lab, we cloned, expressed, and purified a handful of these enzymes and tested them against arabinogalactan as substrates. So I'm going to show you some of the work from Sam in my lab. Sam Benedict worked on the Nocardia enzyme. And what we can see quite clearly is that all of these enzymes, so the 3 from Disgonomonas gattii, this mycosinbacter is a predator of mycobacteria, Nocardia is a closely related bacterium to TB, and they all of these enzymes produce monosaccharide arabinose. This tells us they're chewing down the ends of those glycans with an exoactivity. Sam also went ahead and solved the structure of the nocardial enzyme, and consistent with what we saw in the, it's a little bit different from what we saw in the PDB of that release structure from the Structural Genomics Consortia, but they're really, really quite distinct from all known glycoside hydrolase families. They form this obligate trimer, or in the case of some of the other ones, obligate hexamers or even higher order oligomers. And really kind of interestingly and unusual for glycoside hydrolase, the active site of the enzyme is formed at the interface of each pair of protomers. So there's 3 active sites in this enzyme, for example. The hexamer variants have 6 active sites, et cetera, but they require contribution of residues from 2 protomers to be active, which is really quite unusual in these enzymes. So it's a new structural class for glycoside hydrolases. We're now investigating how they're involved in recycling of arabinogalactan components in some bacteria. And actually, there's another interesting paper that came out around the same time from this group that suggests that they are ancestrally related to phage capsids, which gives kind of an interesting insight into their structure, broadly speaking. Okay, so shifting gears really quickly for the last few minutes of the talk, is this other DUF family I want to dive into, and as a mycobacteriologist, this is really close to my heart, the DUF4 and 8-5 enzymes. So we, again, same idea, we cloned, expressed, purified DUF45s from a range of bacteria. So there's the two D-arabinan ones, there's a xanthus enzyme, one from a phage that Gordonia, again, is a species closely related to mycobacteria, Mycobacterium abscessus, and all of these enzymes, based on our HPLC analysis, give a pattern consistent with endoactivity. So these enzymes, in contrast to the previous family, are cutting within the glycan chains of the D-arabinan, right? And we did other work that I'm not going to talk about that shows they're cutting these actual backbones, the alpha-1,5 linkage. So this is the first ever family of enzymes shown to have this. We believe quite strongly these are the family of enzymes that were discovered back in 1972 and have popped up in mycobacterial literature ever since then without ever actually being identified. Rob Hurd at Newcastle did some phylogeny for us, and he showed that actually all mycobacteria have 2 copies of these enzymes at least, some have more. They're in highly synchronous locations in the genome, so we've named them ERA H1 and ERA H2. Sam in my lab solved the structure of one of these enzymes, although we're missing some bits. This is our the ERA H2 family member, RV3707C, and we learned a few things from that. We learned that they are beta propeller folds. We were able to assign the catalytic residues and show that it's a retaining, enantiomer retaining enzyme. And we also know from our HPAC analysis that actually ARH1 and ARH2, so if you look at these peaks down here, they're quite distinct. ARH1 and ARH2 have slightly different substrate specificities and preferences, and this implies they have different professional roles in the biology of the bacteria, which is work we're now just trying to embark upon. So just real quick before we take off, we've got some new enzymes. What about new biology to go with those enzymes? We're doing a bunch of things right now. I just want to introduce one tool we're using. So we're using high throughput phenotypic screening. And the way we do that is we take our wild type and our mutant, we pin these in high numbers of replicates, like 96 replicates on an individual plate. So Rudy Sullivan in my lab did this work. We can then use software to quantify the area of the colony, and then we can use that as a proxy for fitness. And this can give us really in-depth information about how the bacteria fit, because what we can do is introduce different stressors in the media and then watch how mutants respond to those stressors. And so this is a really nice tool for us. But one of the reasons I wanted to show you this is that this tells us, because there is a small fitness defect, that the bacteria actually really like having these enzymes and they're important for their biology in vitro, although not massively important, right? They're not dying. In contrast, the same enzyme, RV3707C in TB, was shown in an unbiased screen by Miriam Bronstein's group several years back now, that actually when you infect macrophages, the mutant for this gene, you completely abolish, or more or less abolish, the ability for these bacteria to replicate. So there's a really important role for this enzyme during infection that is maybe not quite so obvious in the lab. And so we're really excited about the potential for this, because it means these may in fact be druggable targets, although we're mostly just interested in what they're doing in the cell. So just a quick overall summary. We've discovered enzymes that catalyze release of key cell wall structures in mycobacteria. These, so far, we've got lots more to come, are the DUF2961 family, which are exorhabinofuranases, the DUF25 enzymes, which are these endo-acting enzymes. We know these have important but not yet fully determined roles in mycobacterial biology. And again, we have lots of interesting data there that's not quite ready for prime time, but really exciting directions to come. And I really want to emphasize this, because this is really, Liz really opened my eyes to this idea that the human gut microbiome can act as a great search tool for enzyme discovery. If you have a good idea of a screen you want to do to find a specific activity, really the complexity of the microbiome means the enzymes are probably in there for you. Okay, and last, some acknowledgements. So I mentioned Sam in my lab did most of the work on this project with support by my technician Abby, and Aaron, who's another PhD student lab. Andy Lovering helped us out with some of the trickier aspects of structural biology. I didn't mention the work with Josh at all. Liz's lab in Newcastle, of course, we're 50-50 partners in this project. And so Jen and Omar, who did a lot of the work, Joe, Javier and Jose, and then some other folks who've helped us out, Laura Kiesling at MIT and her, at the time, PhD student Victoria, Spencer in Melbourne, Matthias in Newcastle, Bernie and Robert, and of course the funders, and thank you for your time. And I'm sure Alan and I would be happy to take any questions.
Thank you very much, Patrick, and thank you to both the speakers. With that, we can now move on to the questions. If you have any more questions, please feel free to type them in and to indicate which speaker are they for. So we can start with the first question from Catriona. It's a question for Alan. Did you say that the 3 sulfatases that you spoke about in more detail, including BT1636, are found on the bacterial cell membrane, or did I mishear that? Are the other sulfatases you identified and characterized also secreted, or does further de-sulfation of the colonic sulfomucin occur within the bacterial cell?
As far as we are aware, most, based on predictions and some whole cell assays which we haven't published, most of the sulfatases are in the periplasm. Most carbohydrate degradation occurs and desulfation occurs in the periplasm. 1636 is, we believe, unique in that it is membrane anchored at the cell surface. Our current hypothesis is that because it's the first enzyme to act on those sulfated chains, that it probably needs to desulfate the oligos before they're transported. So we think it's a transport issue. And without transporting the glycan across, the system that senses them will never see them. So it's effectively, the blood is now blind to the presence of significant mucin levels.
Thank you very much, Katrina. We hope that this answers your very good question. And if not, please feel free to follow up. So following on from that, Alan, obviously you mentioned the compound screen and the approaches you're taking to sort of utilize this as a therapeutic strategy. What do you think they will have uniquely to offer as therapeutic target compared to, you know, common antibiotic targets that we have at the minute?
Well, they're more tailored and specific. So if you go, for instance, if you treat with broad spectrum antibiotics, you wipe out significant parts of the microbiome. So any beneficial characteristics you have from that will be gone. And the idea is not to eradicate the species from the microbiome, but is to just restore that balance. So these drugs could be essentially be titratable. And not all IBDs are going to be, inflammatory bowel disease, going to be treatable, driven by carbohydrate sulfatase. But what we're hoping for, the ones that are, will be able to develop drugs to maybe different sulfatases and target, you know, tailor the drug treatment to the severity of the disease. And you can also have, you know, you take, it could be a pill you take when the symptoms spike and you go off it when the symptoms are low. We're hoping to, these carbohydrate sulfatases are incredibly specific. We've already shown that there are very specific compounds that are targeting different subfamilies. So we've got a great opportunity here to target specifically key enzymes in that pathway. And just as a side note, there are some really effective covalent inhibitors of S1 sulfatases. We've got data now showing they only work against steroid sulfatases. They don't work against carbohydrate sulfatases, despite them being in the same family. So there are literally no effective inhibitors of carbohydrate sulfatases. And the current thinking in the field that aryl sulfamates will switch over from the steroid ones to the carbohydrate ones is currently incorrect.
Thank you very much. And Patrick, you showed, and I have to admit, as a complete outsider to the field, I was actually really quite surprised by how little understanding we have of the proteins in, you know, what is a well-known and important pathogen. Do you think this is one of the reasons why the mortality is so high? Is it that there just isn't enough knowledge to be designing drugs well?
Yeah, I don't. That's, I think, how do I phrase that without putting myself out of a job? I think so. There are lots of aspects of microbacterial biology we surprisingly don't understand very well. The biosynthesis of those structures I talked about is really well understood, partially because they are all targets of frontline antibiotics, right? So if you get TB, you're going to take isoniazid, ethambutol. Those target the synthesis of mycolic acids and arabinogalactan, respectively. You might take a beta-lactam, though they're not frontline. That targets the synthesis of peptidoglycan. So we know lots and lots about those synthesis steps, and I think a lot of work in TB is very centered on known or existing drug targets. And there are definitely lots of very dedicated people working on areas outside of that. But I think moving into new areas of tuberculosis biology, does obviously have the potential to open up new drug targets, but also just help us to understand these bacteria better. One of the things I didn't talk about is that a side effect of Liz and I discovering these enzymes is now we've been able to put together the first ever enzymatic lysis tool for mycobacteria. So you used to have to always bead or sonicate these bacteria to crack them open. So now that we can do this enzymatically, this opens the door to kind of new generation diagnostics. So it's by, and that was not something we went into it looking for, expected to happen, but by looking at these new and different areas of biology, they open the door to serendipity, to finding things you didn't expect.
No, that's fantastic. And I guess following on, in a similar line of thinking, it's been ages since I did my microbiology degree, but I remember that mycobacterium was always one of those that, a lot of difficulties also came from the fact of how difficult it was to culture and how incredibly slow did it grow. Do you think that, the better understanding that you might have of the enzymes it depends on, and also on the substrates that it depends on, could improve the way you plan media and the way you plan experiments so that actually it can become an easier bug to work with?
So yes and no. So there's definitely been quite a lot of work on trying to grow mycobacteria faster, full stop. I think there are some labs where that's the postdoc project that they start new people on, and eventually someone gets sick of it and moves away from it because it's not been an easy road. But one of the things that we have, so there's this long-standing idea in mycobacterial biology that there are some sort of quorum-like effects with mycobacteria, and something about being amongst more mycobacteria makes them grow faster. And the mechanism behind that's poorly understood. We have some data that points to some of the enzymes that we're working with, not the ones I spoke about today, but enzymes that act on similar structures that might stimulate outgrowth of the bacteria. And so it could be that cell wall fragments trigger that quorum-like response, in which case, yeah, we could eventually maybe find ways to stimulate their growth, which again could have a bunch of interesting consequences.
That's fantastic. Thank you. And this actually leads me very nice to the closing remarks. Because just to kind of highlight the strength of the talks we had today, I think it really showcases how biochemistry is so much more than we often give it credit for, and how both of the speakers today utilize biochemistry to be able to not only understand more how each species itself behaves, but also how species interact with one another and how bacteria interact with each other as a community. And I think classically, that's not often what you would think that the biochemistry can deliver. So it's important that we have this appreciation. So I would like to thank both of our speakers today, but also all of you who have attended and all of you who have joined the conversation. And you can continue this conversation online by following the Biochemical Society on Twitter. So it's @BiochemSoc and @PPP Publishing, and you can obviously tweet at our speakers or tag them in if you have any comments. We also would like to highlight that we welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. If you have an idea for a webinar in 2023, we invite you to submit a proposal for it. And you can find more information about this on their website, biochemistry.org, where you can also watch recordings of the previous webinars that might be of interest to you. All of the upcoming online series are listed on the website as well. And if you have missed any of the over 60 webinars that have already happened within this series and you would like to watch them again, you can just visit the website or the Biochemical Society YouTube channel, for which the details actually are on the screen, and you can see them here. The recording from today's webinar will also be available to watch within the next couple of weeks. We would like to also invite you to join us on the 27th of July at 2 p.m. British time for the next webinar in this series, Antimicrobial Resistance, the Next Pandemic, when our invited speakers will be discussing approaches required to combat AMR and exploring the roles of different players from the R&D community all the way to policy makers. So this promises to be a very exciting seminar and really good follow-up onto the seminar that we've heard on today. And finally, I would like to highlight that it's more important than ever to stay connected and engage with your fellow molecular bioscientists. Join the Biochemical Society's community of researchers and specialists and take advantage of the key benefits, including discounted registration fees for our society courses and meetings, exclusive access to a wide range of grants and bursaries, especially here very many bursaries for PhD students and for other students to be able to carry on summer lab projects for undergraduates, as well as for PhD students to attend conferences. I really encourage you to look at that, as well as personal online access to 2 of their journals and more. You can visit the website to find out more. And thank you all for being with us today, and hopefully seeing you all in 2 weeks' time for the next seminar. Thank you to the speakers.
Thanks, Urszula.
Yeah, thanks a lot.